The discovery of how proteins are synthesized required the development of cell-free extracts capable of making proteins from amino acid precursors as directed by added RNA molecules. These were first effectively developed in 1953 by Paul C. Zemachnik and his collaborators. Key to their success were the recently available radioactively tagged amino acids. They used these labeled amino acids to mark the trace amounts of newly synthesized proteins. So if the amino acids were radioactively labeled and these labeled amino acids are incorporated into proteins, the proteins can be marked. Early on, the cellular site of protein synthesis was pinpointed to be ribosomes. It was proposed and it was came to know that the protein synthesis takes place at the ribosomes. Several years later, Zemachnik with his collaborator Mahlon B. Hogland made an important discovery that prior to their incorporation into proteins, amino acids are first attached to what we call now as transfer RNA or tRNA molecules. So before entering into proteins, these amino acids are attached to tRNA molecules. Transfer RNA or tRNA accounts for some 10% of all the RNA present in a cell to nearly everyone except Crick. This discovery was totally unexpected. Why this discovery was not unexpected for Crick? Because he had previously speculated that his proposed adapters might be short RNA chains because their bases would be able to base pair and read the appropriate groups on the RNA molecule that served as the template for protein synthesis. So on the tRNA there are some specific bases present, and these bases can read some compatible bases on the mRNA or adapter or template RNA, and they can base pair with these template RNA bases. The transfer RNA molecules of Zemachnik and Hogland are in fact the adapter molecules which are postulated by Crick. Each transfer RNA contains a sequence of adjacent bases. There is a sequence of bases which is called the anticodon that binds specifically during protein synthesis to successive groups of bases, which is called codon, along the RNA template. So there is a specific sequence of bases on the tRNA molecule, and this specific sequence is called anticodon. And similarly, there is some specific sequence of bases on the template RNA, and this sequence on the template RNA is called codon. So anticodon and codon can identify each other, and they can base pair. So here you can see this is a tRNA molecule. It has one attached amino acid. This is the adapter stem where the amino acid is attached, and here the sequence of 3 bases. This is anticodon, and similarly this is mRNA molecule, and this mRNA molecule will act as template for protein synthesis. And on this template RNA there is also a specific sequence of bases, and this specific sequence is called codon. So in this way, the anticodon present on the mRNA identifies a specific codon on the mRNA and binds to this codon.